Hello, students. So, as you may have heard, I am a science teacher, and as such, I would be remiss if I didn't teach you how science works. So, maybe you want to be as cool as Neil deGrasse Tyson, maybe you want to be as neat as Bill Nye. To do so, you use the scientific method, and let's learn how that works. One of the first things that you really need to learn is the difference between observation and inference. Observation should come first. It's really what you want to take when you are taking data during an experiment. And it also starts the whole scientific method. So let's learn what the difference between observation and inference is. Observation is always based on facts. You're going to use your five senses. What you actually see, what you actually smell, what you actually feel. These are observations. Inference is where you use your brain to interpret those senses. So what you think you're seeing, what you think you're smelling from your past experience, it occurs inside your brain. That's why it's inference, inside your brain. And there are two categories of observations. There's qualitative and there's quantitative. Qualitative is like something that's red, it's large, it's hot, it's smelly. These are qualities of something, not actually something that's hard numbers. That's a quantitative measurement, or quantities. 28 grams of salt. The building is 3 meters tall. It's 100 degrees Celsius. That means it's boiling if it's water. Very, very hot. So, difference between observation, inference. So let's practice this a little bit. You see this man, or we think it's a man. We see this picture. Let's look at this picture and let's think of some observations versus inferences. So an observation might be that there is a person wearing blue. An inference could be that the man likes blue. It's not an observation that he likes blue just because he's wearing all blue clothes, outside of his shoes, of course. It's just an observation. You use your eyes to see that the man is wearing blue, or the human is wearing blue. It could be a woman as well. Another one is a human is laying on the stairs. You can see this. He could have fallen down the stairs and dropped his stuff. He could be doing some strange art project. Perhaps he was sleeping there and he just likes to arrange his objects strangely. Of course, the obvious inference is that he fell down the stairs. But we don't know this. That's an inference. It's what we've concocted in our brains. What we can actually see is that there's a human laying on the stairs. So let's talk about the scientific method. Something you may not know about me, I keep bees. By beekeeping, I can get honey. It's an interesting hobby. I got into it because of my family. Specifically on my wife's side, has been doing it for a while. In fact, my brother-in-law is doing an experiment this summer using the scientific method. So bees in hives, when you're keeping them, the hives look like this. They have these frames that he's holding up, and there's usually 10 frames wide. And he noticed, this is an observation, he noticed that they weren't filling the frames all the way up, all the way to the outside. They're not putting honey in there, and he didn't know why. So he did some research. He talked to other apiarists, that's someone who keeps bees, talked to them, talked to a club. He looked online, he read some books, and found out that bees like to have a smaller hive than perhaps this 10 size is ideal for. So he wanted to use a smaller hive and see if that would help the bees make a little bit more honey. So he formed a hypothesis. The hypothesis was, if I have a smaller hive, if I go to 8 frames instead of 10 frames, then I will have better honey production from my bees. And the hypothesis needs to be testable. He was very specific. He said, if I go to 8 hives, then I will have more honey per the weight of the honey at the end of the year. So you have to extract the honey, and then you weigh it, and if you get a heavier weight, that means you have more honey. So that's testable. He can actually see whether or not he gets more honey or less honey than the year before. And it could be disproven. He could find out he has less honey than the year before, but he's not wrong. The hypothesis won't be wrong, it'll just be disproven. So he wanted to do lots of experiments, and he actually has 8 hives that he's trying this in. You don't want to just do one, because that can be a fluke. So you do it with several different experiments, either several trials over and over again, or several at the same time, several experiments at the same time. 8 hives. And you only test one variable at a time. He went to this 8 hives. He didn't add different flowers. He didn't change all the vegetation in the area. He didn't water the flowers around it more. He just changed this one variable, went from 10 down to 8 hives and kept everything else the same. So at the end of the summer, we will harvest this honey. We will take it all out of the boxes, and we will see whether there's more, and we'll look at that data. We'll graph it, we'll compare it to the year prior where we have all these numbers, and see how they compare. That's analyzing the data. Then you come to a conclusion. Okay, I have 150 pounds this year versus 125 pounds last year with the same number of hives. I can now come to the conclusion that my hypothesis is proven to be correct. And then you report it. He will report it to the bee club, other people. He'll report it to me, another beekeeper. And if it's proven, you want to form a theory. You want to conclude, okay, then I should go with this 8. 8 hives is ideal. If it's disproven, then you start over at 1. You make another observation. Okay, what else could be happening? Am I not building enough because they don't have enough of the plants they like? They don't have enough clover. Some other pollinators, a wasp or something, is interfering, something like that. Now, you would start over. And then you publish the results. If he has lots, he's done it for several summers, he's got ideal honey conditions, then he could publish the results in Animal Behavior or some other journal where a lot of people are going to read it, and that's going to help out the scientific community. This is the scientific method. So let's talk about the parts of an experiment. So there's a standard for comparison. That is something that doesn't change. It's something that's as close to normal as possible, so you can actually see what is causing the change in the experiment. So the control in his experiment before would just be 10 frames. Using 10 frames again, maybe this summer was better than last summer. And he does have one hive, a 9th hive, which is 10 frames. That's his control. Then there's an independent variable. This is the variable that you change. You change the number of frames. That's the independent variable. That's the one you're testing, and that, when you graph it, is going to be on the x-axis. The other one is the dependent variable. That's the amount of honey. It depends on the independent variable, and that goes on the y-axis. The last thing, these are constants. So, in these experiments, between the control and all the other hives, he doesn't change the vegetation. He doesn't change how high it is. He doesn't change how often he checks on them. He wants to keep everything as constant as possible. And we'll practice this so you learn the difference between a constant and a control. And a good way to remember this, if you have a graph, we have our y-axis up top and our x-axis on the bottom. Y, you can think of a y because if you put a little line here, you will get a y, so you know it's the y-axis. You remember where the dependent and the independent go, you can think of die. It's die. Dependent on the y and independent on the x, because everybody wants graphs to die. So let's think of an example experiment to see, maybe what type of breakfast would make your biceps grow the fastest. So you would have, if you wanted to do this experiment, first you would want to establish a control. So if you wanted 4 of your friends, they all wanted to see who could grow their biceps the fastest. They would all eat a standard breakfast, maybe for 3 months, just so everybody gets used to that breakfast. Everything's constant. And then your control person, your 4th friend, would just keep eating that breakfast. That's a standard breakfast. And then the 3 other people involved could eat donuts, could eat fruit, and could eat eggs. That's the only thing that you're going to change, is the type of food that they ate, and then you will measure how big the biceps grow. And so your constants are, all the meals are going to be the same. For lunch and for dinner, it'd be very hard to do, but this is what you'd have to tell just to figure out how breakfast affects your biceps. You want to be the same age. You want to be the same gender. If you're male, you're female. You may have different, or you will have different hormones, which might affect bicep and muscle growth. And you also want to measure at the same time. And another big one would be maybe the same exercise plan. If someone's doing more exercise, their biceps are probably going to grow differently or more than someone who's doing less exercises. Those are all constants. The control is the person that has everything else, that has all of the constants. But the constants are each individual item that must stay the same between everybody involved in the experiment. So here is graphing the results. This is how you would analyze the results. And say after 3 months, you were 3 months on the same breakfast and then 3 months on each individual breakfast, you could actually see the effects of the experiment as long as you actually kept all those constants the same and constant. So your control person, you could see, had 30 centimeter bicep circumference, and that's about average for your age. The donuts, you could see, has a smaller bicep circumference. The fruit has a little bit higher, and then the eggs have the highest. And then you would maybe publish your results, and you might do research to see why this might be. You might notice that eggs have a lot more protein in them, but this is what your graph would look like. This is proper format. And you will see our dependent is on the y-axis. That's what we measured, the bicep size. And you guessed it, the independent is on the x-axis. All of these things, these are what we changed. We changed what was eaten for breakfast, so that's the independent axis. Another thing to note is that there's a properly formatted title up here. You always want to have a figured number if you're reporting these things, and information that tells you about what the actual experiment was. So don't just say breakfast foods. You want to say bicep size after 3 months versus breakfast. That way we get all of the information in the actual figure. And also notice that we have quantitative information here. One way to tell that it's quantitative is we see units. Don't forget your units. I know you've had this in other science classes. Don't forget your units. That's how we know it's a quantity. And then what do we have in our independent axis? Well, actually, we have qualitative information. There's not a number involved here. This is just a quality, the quality of the breakfast. So it's qualitative information. So here's some practice, and you can hit the pause and practice labeling the variables, the constant, the control, and the results in this example. So Susan is always late for English. The deans will not give her a 1-minute pass. They don't feel it's necessary. She must find a better way to get from dance by the gym to the upstairs. She usually uses the front hallway K and the stairs by hallway K. She experiments over the period of 6 days. She tests the hallway J for 3 days and hallway L for 3 days. She wears the same shoes, carries the same books, and stops at her locker each time. She finds the hallway L cuts off 3 minutes of her time. So go ahead and pause here, figure out what the variables are, the constant, the control, and the results. Okay, so let's go over each of them. There are two variables. Our independent is the hallway. This is the thing that you can change. And then the dependent is the time, the amount of time that it takes. It's the thing that she measures. Another thing is the constant. So let's look at the constants. Those are the same shoes. Anytime you see same, you know constant. So same shoes. She carries the same books. The weight might throw her off. And she stops at her locker each time. Another thing would be that in between the two tests, she has the same number of days, 3 days. That is a constant. The control, what do you think it is? Yeah, it's the original hallway. So she knows after repeated experimentation before, the control is the original hallway. This one was the tough one. And then the results, I think, are pretty obvious. She finds that the hallway cuts off 3 minutes of her time. This is great information to have, and she might report this to her friends who are also having problems getting to class on time, because everybody knows how important it is to get to class on time. So another important thing to notice is historically, not all scientists have done it this way, and not every single scientist practices this exact method. This is a great method that incorporates every important procedure, which is why we're going to practice this method, but it's not always the case. For instance, Thomas Edison created the light bulb just by pure experimentation. There wasn't information out there for him to really figure out how a light bulb worked, so he just had to try a whole bunch of different things, lots and lots of different experiments, until he figured out how that filaments actually work. Feathers don't work. String doesn't work. You have to use a filament to heat up and get that light bulb to work. And then there's accidental discoveries, such as Alexander Fleming discovering penicillin. He just happened to leave his fruit out to rot and find out that it killed all of his bacteria plates, and that's how he discovered penicillin works. He did a lot of experimentation afterwards to figure out which part of the mold actually was killing the bacteria, but his initial discovery was just an accident, and sometimes that happens. It's perfectly fine as long as you follow scientific practices afterwards and report it in the proper way, where a scientist can look at it and determine what actually was done, and so that it's repeatable, so that now everybody who needs it can get access to something important, like a light bulb or penicillin. So, thank you guys for listening. I hope you got great notes on this, and we will talk about this tomorrow.